Today we are going to start carbohydrates. Carbohydrates are most abundant molecules in our body as well as in nature. They have carbon, hydrogen, and oxygen. So they are called. They are also known as hydrates of carbon. The carbohydrates may be defined as a polyhydroxyaldehydes or ketones or compounds which produce them on hydrolysis. So the common formula of carbohydrate is CH2O power N. Now functions of carbohydrates. As we know, the most abundant dietary source is carbohydrate, which gives energy, and it acts as precursors for many organic compounds such as fats, amino acids. Carbohydrates are seen in all cell membrane and also participate in cell functions such as cell growth, adhesion, and fertilization. Carbohydrates are major source of storage energy, that is like glycogen, glucose, etc. The term glycome refers to entire spectrum of carbohydrates of an organism. Glycomics is a study of glycomes or glycobiology or carbohydrates. Now, classification of carbohydrates. Carbohydrates are often referred as saccharides. Now, carbohydrates are divided into three types: monosaccharides, oligosaccharides, polysaccharides. Now, monosaccharides, in this we have two types like aldose and ketose. Aldose group which is attached to the molecule is aldose, and ketose is where the ketone group is present. And now in oligosaccharides we have disaccharides and trisaccharides, tetrasaccharides, etc., and so on up to 10. 2 to 10 molecules present in a carbohydrate is known as oligosaccharide. So they are named according to the number of molecules present, units present in the chain. Now polysaccharides, they are divided into homosaccharides and heteropolysaccharides. Homopolysaccharides and heteropolysaccharides. Now here you can see the monosaccharides. In monosaccharides we have triose, tetrose, pentose, hexose, hectose, etc. Now triose have number of carbon is 3, and we always know the monosaccharides are divided into aldose and ketose. So the triose aldose monosaccharide is glyceraldehyde, whereas ketone group is deoxyacetone. Now tetrose 4 carbon atoms. Aldose exithrose is present in aldose, whereas in ketose we have erythrulose, whereas pentose we have 5 carbon atoms. Aldose group has ribose and ketose has ribulose. Then hexose 6 carbon atom like glucose, galactose, mannose, and in ketose we have fructose. Now hectose we have 7 carbon atom. Examples pseudoheptose, and ketone group has pseudoheptulose. Now let us go in detail. Monosaccharide. The common formula is CnH2ON. It is also called as simple sugars. They cannot further hydrolyze. So we have two groups as we see in aldoses and ketoses. When the functional group is aldehyde, that is this is the functional group or aldehyde group. It is attached to the carbon, so it is known as aldoses. And example is glyceraldehyde, glucose, etc. Now ketoses, when functional group is ketone or carbonyl, it is known as ketoses. This is a functional group of ketose, and example is dihydroacetone and fructose. Now let us know about the common monosaccharides. The first most common monosaccharide is glucose. It is the main component of dietary polysaccharides, that is starch, and it is an aldohexose. See, we have aldohexose. Aldo means it is the aldehyde group. The monosaccharide is under aldose, and it has hexose, so it has 6 carbon atoms. Now fructose. Fructose, the sugar present in fruits is known as fructose. Mainly it is present in fruits, honey, and a component of table sugar, and it is a ketohexose. So we can remember glucose is an aldohexose, whereas fructose is a ketohexose. It is under ketosis, and the number of carbon is hexose 6. Now galactose. The compound of milk sugar, lactose, is known as galactose. Galactose and mannose are components of glycoproteins and mucopolysaccharides. Amino sugars. When one or more hydroxyl group of monosaccharides are replaced by amino group, they are formed as amino sugars. Example: D, D-glucosamine, D-galactosamine, etc. And they are present in heteropolysaccharides. Now deoxy sugars. These sugars have one less oxygen compared to the parent molecule groups. Like CHOH and C2OH become the oxygen is removed here. Like we have one oxygen less than the parent. This is the parent, whereas the oxygen is removed and it becomes like this, CH2. And this is the parent, CH2OH, where oxygen is removed and becomes CH3 due to absence of O2. So D2 deoxyribose and D-ribose are important sugars and they constitute DNA and RNA. So deoxy sugars are very important. Now L-ascorbic acid, it is also known as vitamin C. The formula is C6H8O6. It is water soluble vitamin which resembles the structure of a monopolysaccharide. Here is the structure of the ascorbic acid or vitamin C. Now trioses like glyceraldehyde and deoxyacetone are seen as a metabolic intermediate. Now after monosaccharides we'll go to oligosaccharides. Oligosaccharides have 2 to 10 monosaccharide molecules in them, and based on number of monosaccharides present in, they are divided as disaccharides, sidesaccharides, tetrasaccharides, pentasaccharides, etc. and so on up to 10. They are seen as part of blood group antigens. Now disaccharides. In oligosaccharides, the most common is disaccharides. Disaccharides holds 2 monosaccharide molecules which may be similar or dissimilar, and they are attached through glycosylic bond. Now let us understand glycosylic bond. Here we can see the glycosylic bond forming by OH and H, and water will be formed by combination of these two, whereas they leave an oxygen. For example, here you can see this is a glucose molecule, this is a glucose molecule which is monosaccharide. Now 2 monosaccharides are combining together, that is disaccharide. Now this OH will react with each other forming water, and after forming water we will be left with one oxygen which is left here. So forming of this bond is known as glycosylic bond. Now here disaccharides are crystalline, water soluble and sweet to taste. These are of two types, reducing disaccharides as well as non-reducing. Reducing are the disaccharides which have free aldehyde or ketone group, for example maltose, lactose, etc. And non-reducing are with no free aldehyde or ketone group like sucrose, tetrahydrose, etc. Now let us see about common disaccharides. The first one is maltose, which is a reducing disaccharides. That is, it has an free aldehyde or ketone group. Now maltose is made up of 2 alpha D glucose units held together by alpha 1,4 glycosylic bond. The first carbon of first glucose unit is free, so the maltose is reducing disaccharide. And maltose can be hydrolyzed by dilute acid or the enzyme maltase to liberate 2 molecules of alpha D glucose. Here alpha D glucose is held by alpha 1,4 glycosylic bond. We know what is glycosylic bond, but what is alpha? We'll know what is alpha and what is 1,4. Here we can see the bonding between 2 glucose molecules, that is 2 monosaccharide molecules forming a bond, glycosylic bond. Here alpha means the bond is connected side by side, so this is known as alpha. And 1,4 is the first carbon of first glucose molecule is attaching or bonding with 4th carbon of the second glucose molecule. So alpha refers side to side. 1,4 is attachment of first carbon to 4th carbon. Then glycosylic bond is forming a bond with O while H2O is released. Now let us go to sucrose, which is non-reducing sugar or disaccharide. It is also called a stable sugar or cane sugar that is used in everyday tea, beverages, etc. And it is mostly produced by sugarcane or sugar beets. And sucrose is made up of alpha D glucose and beta D fructose held by alpha 1 to beta 2 glycosilic bond, that is alpha and beta. Here beta means the formation of bond is like they are from up to down, or they are stacked to form the bond. This is known as beta arrangement. Here sucrose is made up of glucose and fructose. So glucose plus fructose gives rise to sucrose. Now lactose. Lactose is reducing disaccharide. It is commonly called as milk sugar. It is made up of beta D galactose and beta D glucose held by beta 1,4 glycosilic bond. Lactose of milk is most important carbohydrate in nutrition of young mammals. Lactose is hydrolyzed by intestinal enzyme lactase to glucose and galactose. Now glucose and galactose together form lactose. Now let us know other molecule which is known as isomaltase. Then when two glucose units are joined by alpha 1,6 linkage glycosilic bond, the disaccharide form is known as alpha maltase, and it is a byproduct formed during the digestion of starch by alpha maltose along with the glucose and maltose. Now sucralose. Sucralose is also known as artificial sweetener. Now sucralose is trilorosucrose is which is about 500 times sweeter than the normal sucrose. These are not digested, hence no calories are absorbed in the body. And most common other sugar substitutes are like saccharin and cyclamate. Now Benedict's test. Benedict's test is used to test the reducing sugars. So if reducing sugars like maltose and lactose are tested, then it gives positive results, whereas sucrose is negative as it is non-reducing. Now invert sugars. The sucrose is called as invert sugar as the rotation changes from plus 66.5 degree to minus 19.8 degree from dextro to levoretirate on hydrolysis. Now polysaccharides. Polysaccharides are polymers of monosaccharides which yield more than 10 molecules of hydrolysis. See example C6H10O5. Now polysaccharides are of two types: homopolysaccharides, heteropolysaccharides. Homopolysaccharides, they are made up of only one type of monosaccharide units like starch, cellulose, glycogen, dextrin, dextron, etc. Now let us know some common homopolysaccharides. First is starch. It is a homopolymer composed of D-glucose units which are held by alpha glycosilic bond. It is also known as glucosan or glucan. The starch has two polysaccharides, that is water soluble amylose which is 15 to 20% and water insoluble amylopectin which is 80 to 85%. The amylose is a straight chain with 200 to 1000 D-glucose units which are held by alpha 1,4 glycosilic linkages. Now amylopectin is branched with alpha 1,6 glycolinkages and amylopectin is branched, but the straight chain composed of alpha 1,6 glycosilic bond, and where the branching occurs, where the branching occurs, it is attached by alpha 1,4 linkage. Now 20 to 30 D-glucose units per branch we can see in this amylopectin. The starch is digested by alpha amylase produced by salivary glands and pancreas. Now let us move to second example of homopolysaccharide, that is glycogen. The carbohydrates reserved in animals are also called as animal starch. So it is also known as animal starch. It has similar structure with starch but packed more densely. The chains are arranged in concentric manner and are branched inside and unbranched outside. They weigh up to 107 daltons and diameter is about 21 nanometer. Liver and muscles are main sites of glycogen storage. Now let us move to cellulose. Cellulose occurs exclusively in plants. Cellulose is composed of beta-D glucose units held by beta-1,4 glycosylic bonds. Even cellulose is not digested. It is major component of fiber which helps in decreasing in absorption of glucose, cholesterol from intestine and helps in relieving constipation. Now chitins, the homopolysaccharide made up of N-acetyl glucosamine monomers found in protective outer skeleton or exoskeleton of insects and crustaceans. They have like cockroach etc. They have chitin. Now insulin, the polymer of fructose, that is fructose and D-fructose. It has no dietary importance. It is used for inulin. It is used for determination of glomerular filtration rate in renal function test. It is only used in that way, but it has no function. Now agar. Agar is seen in seaweeds made up of repeating units of galactose. Agar has high water holding capacity. The sulfated agar is used as laxative, whereas purified agar is used as bacterial culture. Now dextrins. Dextrins are incompletely digested products of glycogen and starch from the amylopectin. These are finally completely digested to maltose and glucose. Dextrons. Dextrons, they have high molecular weight. Dextrose other name is D-glucose. Now heteropolysaccharides or mucopolysaccharides, they have different types of sugar or their derivatives, also called as heteroglycans, usually made up of amino acids and uronic acids. Amino sugars usually is an acetyl glucosamine or N-acetyl gallosamine. Uronic acid usually is glucuronic acid or ichloronic acid from glucose. Due to their amino sugar content, they are also called as glycosamine glycans.